Greetings from University of Missouri, Columbia. My name is Anandhi Upendran. I'm the Director of Biomedical Innovation and also Lead Scientist at MU Institute of Clinical and Translational Sciences. At the outset, I would like to congratulate Dean Hemalata for organizing this international conference on basic and translational cancer research. Thank you very much, Hema, for organizing it, and it's a pleasure to present our work in this virtual conference, and also to forget about this COVID-19 pandemic that is ongoing right now. And I hope all of you are safe. With that, I would like to talk to you about our work on the retinoblastoma. And the topic today I will be talking about is targeting HMGA protein inhibits retinoblastoma cell proliferation. This work has been published in RSC Advances recently. And it's a teamwork, and we did this work with the collaboration from Dr. Professor S. Krishna Kumar from Vision Research Foundation, Shankar Netralayam. I'll be talking about the outline of my talk includes like a brief background about the retinoblastoma, current clinical status and the problems, the solution that we have proposed and the studies that we have carried out, how we did it and the results and the conclusions. Retinoblastoma is a childhood eye cancer, and as the name suggests, retina, it starts from the retina itself. And the cancer develops rapidly from the immature cells of retina from the very back part of the eye, and it's most common primary malignant intraocular cancer that's common in young children and rare in adults. This can be congenital, but most often it is sporadic and non-hereditary. The risks associated with retinoblastoma include vision loss and spread to the distant organs such as brain and bone marrow, and rarely it can be life-threatening also. Retinoblastoma accounts for about 2% of all childhood cancers. In a global perspective, 1 in 15,000 children develop retinoblastoma, and about 8,000 to 9,000 children are newly affected every year. And in a recent report that has been published in Japan Journal of Ophthalmology, it is reported that the average occurrence frequency for retinoblastoma is about 1 is to 16,823 births per year. And of that, 89% of the patients are younger than 3 years and 41% are younger than 1 year. In US alone, about 200 to 300 children who are less than 5-year-old have this retinoblastoma, and more importantly, 90% of the children of these children, means the children affected by retinoblastoma, live in developing countries. The common therapies that are used for retinoblastoma include cryotherapy, thermotherapy, radiation, surgery, and chemotherapy. Cryotherapy involves a freezing of these abnormal cells, while thermotherapy involves use of heat. Radiation is the common one that is being used for most of the cancers that use X-ray, while the surgery involves treatment by removing the eye or a part of the optic nerve. Chemotherapy is the common way that is being used for the retinoblastoma treatment. It can be local or systemic. The local chemotherapy involves treatment procedures using melphalan by an intravitreal approach, while or by means of intraarterial administration of carboplatin. The other systemic administrations involve use of combination of drugs such as vincristine, etoposide, carboplatin, or these drugs by themselves. The drawbacks of these current therapies are such as cryotherapy, thermotherapy. Both these therapies destroy abnormal tissue, but they may cause tissue scarring and swelling, while the radiation results in killing of abnormal cells, while the problem associated with radiation involves partial or complete vision loss. Surgery, as you know, it's not a desired procedure by itself, but they may cause risk of infection or other complications. The most important drawback of this chemotherapy is these chemotherapy that are being administered may be toxic to retina or to the retinal pigment epithelial cells in advanced stages. That may also result in removal of eye or vision loss, and importantly, it's a toxic side effects, drugs toxic effects that's associated with the chemotherapy, which is a big major drawback. Chemotherapy is usually offered for both intraocular and extraocular retinoblastoma. While it can be administered systemic, it can be systemic, conjunctival, or intraarterial or intravitreal. It's mostly used to avoid complications related to radiation or to prevent development of secondary cancers, which means metastasis. The proposed solution or the alternates for these chemotherapy can be targeted molecular therapy that can help to reduce drug toxicity, prevent vision loss, and improve therapeutic outcomes. Targeted molecular therapy will help to change the things at a genetic level by using siRNA or aptamers, which can improve the therapeutic effects significantly. So towards this approach, first we need to identify a target that can, and the second one is to use a suitable delivery vehicle for using this, for delivering this siRNA or aptamers in a proper way without degradation. What is the first target that we identified is a high mobility group A protein, which is called the HMGA2 protein. HMGA is a family of proteins which it's a two non-histone nucleoprotein subsets, HMGA1 and HMGA2, that binds to AT-rich minor groove of the DNA, alters the DNA chromatin structure, facilitates attachment of transcription factors, and it's also function as a differential regulation of the transcription of genes. This high mobility group A protein is always associated with invasiveness and metastasis of the disease, and the unique feature is it's overexpressed only in tumors and not expressed in healthy adult tissues. Through the research of our collaborators, especially Professor Krishna Kumar's group, we have identified that the retinoblastoma tumors overexpress HMGA2 proteins, and it has also been correlated with high-risk histopathology features that involves diffused invasion of choroid and optic nerve. So, targeting this HMGA2 protein, or if we can silence this HMGA2 protein by any means, it would help to reduce the metastasis or reduce the invasiveness or the severity of the disease. That's the whole idea. So now that we know that HMGA protein is involved in the metastasis and invasiveness of this whole tumor. So either HMGA proteins or the mRNA transcripts can be used. siRNA or aptamers or DNA minor group binders such as netrapsin can be used as targets to target this HMGA2 protein. And through our studies, it has clearly been demonstrated that if we target this HMGA2 through an siRNA, it has promoted cell death through apoptosis and cell cycle arrest in cancer cells. Therefore, selective delivery of either an short interacting RNA of HMGA2 or an aptamer to the retinoblastoma cells through cell surface targeting proteins that are overexpressed in cells, it will be very helpful and as a targeted therapy. So what is the cell surface protein? Next is that question. What can be used as a cell surface protein to target this HMGA2? Nucleolin protein that is overexpressed on the surface of the RB cells. We thought that can be, it's a shuttle protein it's a shuttle protein that shuttles nucleolus to the nucleoplasm, cytoplasm, and cell surface, and it can be used, that nucleolin protein itself can be used as a delivery vehicle either for the siRNA or for aptamer delivery. So usually the delivery agents of siRNA or aptamer include antibodies, peptides or nanoparticles, and therefore antibody aptamer pinces, antibody siRNA conjugates are known in literature, and additionally chemically programmable antibodies conjugated to RNA aptamers are also known that are known to improve the affinity. They are known to improve the affinity of the target molecules. Aptamer conjugated with drugs have also been used. However, there are limited reports about the antibody aptamer conjugates. So what we thought as a delivery system is to use a nucleolin aptamer to deliver the HMGA to siRNA, which means nucleolin aptamer mediated delivery of HMGA to siRNA, or we can use the nucleolin antibody mediated delivery of HMGA to aptamer, which means we can prepare nucleolin antibody aptamer HMGA to aptamer conjugate to deliver this HMGA to aptamer. So we have planned. So as I repeat, we have prepared aptamer siRNA conjugate in which the nucleolin is used as an aptamer and siRNA is being delivered. We have also explored the opportunity of using nucleolin antibody to deliver the HMGA to aptamer. So here is the pictorial representation of this antibody aptamer based delivery. So the siRNA is being delivered using the nucleolin aptamer, which is shown here, and the second picture down below shows the delivery of the HMGA to aptamer through the antibody. So either way, we anticipated, the hypothesis is the HMGA2 protein can be knocked off or can be silenced by means of using the nucleolin antibody or nucleolin aptamer, so that HMGA2 is selectively targeted and selectively targeted in a way that it will help to limit or prevent cell proliferation or limit the apoptosis. So as of next step, we synthesized the HMGA2 targeted conjugates, namely the nucleolin aptamer HMGA2 siRNA conjugate and also the nucleolin antibody HMGA2 aptamer conjugate. The nucleolin aptamer HMGA2 siRNA conjugate was synthesized using equimolar concentration of aptamer and siRNA using a sulfo-SMPB bispecific linker, while the antibody aptamer conjugate was synthesized by using the traditional EDC-NHS conjugation. Once these conjugates were synthesized, these conjugates were characterized. The nucleolin aptamer siRNA conjugate was characterized by page gel electrophoresis. We used a non-denaturing page to first clearly prove. We used both the fluorescence. In this case, the HMGA2 siRNA was conjugated with a FIFC dye, and therefore we could clearly show the presence of the conjugate, the nucleolin aptamer HMGA2 siRNA in the fluorescence channel, as well as in the ethidium bromide channel only for the siRNA, and with the third one represents the merged image where we can see the pure nucleolin aptamer siRNA conjugate in lane 8. The nucleolin antibody aptamer conjugate was characterized by UV visible and spectroscopy and also by zeta potential. When we measured the zeta potential, there was considerable difference between the plain nucleolin antibody and the plain HMGA2 aptamer. There was the zeta potential of the conjugate itself was significantly different, from which we could conclude the formation of this nucleolin antibody HMGA2 aptamer conjugate. Further, we also characterized this nucleolin antibody HMGA to aptamer by means of page gel electrophoresis. The presence of antibody was confirmed by Comasi blue staining, while the presence of aptamer was confirmed by gel red staining, and the whole conjugate was confirmed by means of fluorescence imaging. In this case, the HMGA to aptamer was conjugated with the CY5 dye, and therefore the fluorescence was, as you see in the case of this Comasi blue stain, lane number 4 clearly shows the presence of the pure conjugate. In the case of the fluorescence image lane 4, a faint signal has shown here. A faint signal here confirms the presence of this nucleolin antibody aptamer conjugate, while that of the presence of the aptamer in the conjugate is confirmed by the gel red staining. Once we prepared the conjugates, we performed detailed stability studies. We investigated the stability in PBS and 10% FBS for both the conjugates. We showed the stability by performing the non-denaturing page gel electrophoresis for the aptamer siRNA conjugate, while we conducted fluorescence studies to show the stability of the antibody aptamer conjugate. Both UV as well as fluorescence spectral studies spectrum recorded at different time points clearly showed that the conjugate is highly stable and robust. For that matter, both the conjugates are stable and robust in these media. The next step was to confirm the in vitro targeting, which means that whether this nucleolin aptamer is able to internalize whether this whole conjugate gets internalized in the cells or not, especially in the retinoblastoma cells. Whether this aptamer siRNA conjugate, antibody siRNA conjugate can internalize effectively into the cells is the objective of this receptor binding studies. First, let us look into the what we took the very cells, incubated them with the nucleolin aptamer siRNA conjugate along with proper controls. In case of the nucleolin aptamer and HMGA to siRNA conjugate, we used plain nucleolin aptamer as the control. As you can see clearly in this, the nucleus was stained with DAPI, and of the cells, the very cells were stained and incubated with this nucleolin aptamer siRNA conjugate. In the DAPI channel, as expected, we could see only the fluorescence from the DAPI. In the FITC channel, with the nucleolin aptamer, with the use of this nucleolin aptamer alone, we could see the fluorescence, but with the nucleolin aptamer siRNA conjugate, we could clearly see that the presence of this conjugate inside in the nuclear region compared to that of the plain nucleolin aptamer alone. So in the merged channel, we could clearly see that the aptamer siRNA conjugate has internalized effectively into the nuclear region. So this experiment was with a conjugate in which FITC is present. When we look into this panel B, where we have prepared this nucleolin antibody HMGA to aptamer, it's clearly shown from the DAPI channel. In this case, we had conjugated the HMGA to aptamer with the CY5 unit, and it is clear that in the DAPI channel we couldn't see nothing is expected, and as such only the DAPI fluorescence is seen. But we compared the fluorescence with that of the transfected HMGA to aptamer in the cells, and if you can clearly see that the amount of aptamer that is shown in the 50 that has internalized in the cells are very low compared to that of the number of cells that has this nucleolin antibody aptamer conjugate. In the merged channel, it's clearly evident that the nucleolin antibody binds to the surface of the nucleolin protein, and it has there is a possibility of internalization, and it has internalized effectively compared to that of the free HMGA2 aptamer. So these results confirm the binding ability and internalization of these conjugates compared to that of the free aptamer as well as the free HMGA2 aptamers as well as the free NCL aptamers. The next step was to explore the cytotoxicity, or to determine the cell viability, percentage cell viability of these conjugates, percentage when these conjugates were used. So detailed in vitro cytotoxicity was performed using retinoblastoma cells. We treated the retinoblastoma cells with the nucleolin aptamer HMGA2 siRNA conjugate, along with the proper controls such as the plain nucleolin aptamer and the HMGA2 siRNA. Well, in the case of this nucleolin antibody aptamer conjugate, we treated the cells with nucleolin antibody aptamer conjugate along with the proper controls such as the nucleolin antibody itself and the aptamer, along with that of the physical mixture of the nucleolin antibody and the aptamer. So the results are, as you can see from the graph, the results are presented in this table. As you can see here, the IC50 values for the individual components of the individual components are listed here along with that of the conjugate. In the case, if you look at this nucleolin aptamer HMGA2 siRNA conjugate, the conjugate shows a twofold or a reduction compared to that of the individual entities such as the nucleolin aptamer itself and the siRNA itself. So the nucleolin aptamer HMGA2 siRNA conjugate shows a IC50 value of 72, which is twofold lower compared to that of this aptamer alone, and several folds lower than that of the siRNA itself. When we compared the individual IC50 of the individual components of the nucleolin antibody HMGA2 aptamer, it's clearly shown that the nucleolin antibody HMGA2 aptamer is the cell viability or the IC50 is significantly lower compared to that of this antibody, plain nucleolin antibody and the aptamer itself. The percentage of cell viability for the aptamer siRNA conjugate was 47% at 100 nanomolar, while for that of the antibody aptamer conjugate was 41% at 130 nanomolar. Both the aptamer siRNA conjugate and the antibody aptamer conjugate showed significant decrease in cell viability, while the nucleolin antibody as well as the free aptamer themselves did not show any signs of toxicity. However, the IC50 value of the antibody aptamer conjugate was about 90 nanomolar, and we have also determined through our studies that how much of antibody or how much of aptamer is present in this conjugate. So by this 90 nanomolar of this nucleolin antibody HMGA2 aptamer conjugate contains about 26 nanomolar of HMGA2. Based on this calculation, we could figure out that this nucleolin antibody HMGA2 aptamer is approximately 50-fold more cytotoxic compared to that of their individual HMGA2 aptamer itself. So this nucleolin antibody has been used as targeting vector, and it has camouflaged this HMGA2 aptamer, and it has retained the It has facilitated the retention of the structural integrity and the functional property, thereby improving the cytotoxicity in the very cells. The enhanced cytotoxicity of the nucleolin aptamer HMGA2 siRNA conjugate can be attributed to the synergistic effect of the nucleolin aptamer itself and the HMGA2 siRNA, and it's also clearly evident that both the aptamer and siRNA are functional in this conjugate. The enhanced cytotoxicity of the nucleolin antibody HMGA2 aptamer conjugate can be attributed due to the targeted delivery of this HMGA2 aptamer to RB cells. So finally, to conclude our study results, I could say that we have used a targeted approach we have explored at least a targeted approach for retinoblastoma therapy. We have used overexpressing nucleolin shuttle protein as a vector for the delivery of the siRNA as well as the aptamer. Nucleolin antibody HMGA2 aptamer and nucleolin aptamer and HMGA2 siRNA conjugates were synthesized and characterized in detail. These conjugates are highly stable and robust. Receptor binding affinity has confirmed that both conjugates are capable of show receptor binding affinity, and then they can internalize effectively into the cells. Both the antibody aptamer conjugate as well as the aptamer siRNA conjugate showed good cytotoxicity. However, from the studies we could clearly see that the antibody aptamer conjugate has unique and translatable characteristics compared to that of the nucleolin antibody siRNA conjugate, because it's also easier to synthesize, it's scalable and has better conjugation efficiency, has a higher degree of cellular internalization in the very retinoblastoma cells. Most importantly, it has enhanced toxicity, which is 50-fold higher, that has been demonstrated in the very cells compared to that of the free aptamer. With all these important points, it's clearly evident that this nucleolin antibody HMGA2 aptamer conjugate has the potential for exploring for preclinical applications. So finally, I would like to conclude my talk by acknowledging the team members who are vital for completing these studies. As I had initially mentioned, this work wouldn't have been possible without a wonderful collaboration with Professor Krishna Kumar and his team at Vision Research Foundation, Shankar Netralaya. Our team at MU, we had Professor Raghuraman Kannan, Dr. Ajit Zambre and Jack Joe, who had immensely contributed for several aspects of this project. And we couldn't thank enough the Childhood Eye Cancer Research Trust for funding this project, and also we have used the endowment funds from Michael J. and Sharon R. Buckstein Endowment. And finally, I would also like to thank you all for your patience for listening to this talk. Thank you again. And thank you, Hema, for giving us an opportunity again and also for conducting this virtual conference. Congratulations, and I hope it was very successful. And please be safe. Our best wishes. Thank you.